Welcome, everybody. This is my first lecture for the class of genetics, and it's an introductory lesson and lecture. And I started the slide with a picture from a company that is near and dear to my heart. I used to work here at this company. It's called Genentech, and they are a biotechnology company with the purpose of using the tools of genetics to create highly therapeutic drug products to treat illnesses such as lymphoma, other cancers, breast cancer, heart attacks, hemophilia, just a lot of other autoimmune diseases as well. So it's a very cool company. They also make a drug for cystic fibrosis and growth hormone, and all of these things are really, what they do is they do a version of biotechnology that involves using bacteria or mammal cells to get those cells to express a protein of interest, and they take that protein of interest and make it into a drug formulation. So we're going to give a background, a little bit of the history of genetics, and talk a little bit about some of these things. So again, biotechnology near and dear to my heart. So let's go and get started on this. The introduction part first, chapter 1. They start the chapter by providing some information about some of a concept called translational medicine, which is using these genetic findings to directly translate into methods of diagnosis and treatment. And, the more information that we have about our genetics and the Human Genome Project and that sort of thing kind of leads us to some medicines that will translate into cures for diseases, or treatments, I should say, either one. That Human Genome Project is fascinating. It's basically a project that involves mapping all of our genetic information where genes are located in our DNA. And once you know where a gene is located and you know what its function is, you can figure out how to manipulate some things to use it to direct and guide treatment methods. So this coupling of research and drug development, kind of young, it came about in the 70s, and it's really, really grown to be a huge industry. And lately, cardiovascular disease has been a focus of some of these studies, and they talked about, in general, we kind of have really bad cardiovascular disease issues, and one of them is high cholesterol, and research on this gene is a good example of this. There's a lot of others that are noted with biotechnology, but this actual gene, the PCSK9 gene, directs the traffic to create a protein that binds to LDL receptors on the liver cells. And if it doesn't bind, the receptor is returned to the cell surface where it can remove more LDL from the bloodstream, which is really good. So if you can figure out how to prevent this from happening, this binding, you could potentially have a drug treatment, which would be very good for people with high cholesterol. So a little bit of the history. This goes way back, folks. It's back as far as 8,000 to 1,000 BC. So a very long time ago, there was domestication and selective breeding of animals and plants, and what we would call artificial selection, because we're sort of forcing it. What natural selection is, what we'll talk about, where it happens kind of, the selective pressures are more natural. And so anyway, it goes way back into time, and in some ways it's a very old field, and in some ways it's a very young field. But anyway, so Hippocrates, he was kind of involved in Aristotle, both in, if you think of the Hippocratic Oath, that's where this is coming from, but back a little bit before c. 500 to 400 BC, and then 384 to 322 during Aristotle's time, they tried to start to explain heredity, and Hippocrates was the first to sort of explain this concept of heredity and traits. But Aristotle came in and tried to finish the job and explain it a little better. They were not quite right about it. They had some aspects of it correctly, but Aristotle was interesting because he believed, if you read about it in your book, that there was some fact of, he related menstrual blood to this thing that was kind of needed to be heated and nurtured, and it was kind of the physical substance that gave rise to offspring, but they needed that male semen that contained the vital heat to kind of get things going. And so some weird ideas, but neither quite right. Even in the 1600s, William Harvey came up with this theory of epigenesis, and here's sort of a homunculus image over here, which is technically this is a sperm with a fully developed person in there, that his belief was that organisms developed from fertilized egg through the developmental events that transform egg into an adult, which is good. But there was some belief that it was, contained in this thing called the homunculus, which, just interesting ideas that came about. But this one was pretty good. It was pretty close on. Cell theory was very important. It came about in the 1830s, which honestly, folks, is not that long ago. Sladen and Schwann came up with this idea in 1930, basically that all organisms are composed of cells derived from pre-existing cells. And that's huge foundational information relative to biology and the study of biology. There was a concept that was disproved by Louis Pasteur, but it was that things can spontaneously generate from non-living components. This was a big deal at one point. That was the central dogma or the thought process of, it was called spontaneous generation, but there were some studies that kind of disproved that, and Louis Pasteur sort of led the way. But here's where it gets good. In the mid-1800s, Darwin set sail and went and voyaged around the world, and mostly South America, and learned some things about evolution. And what he was missing, though, was what came about later from a monk named Gregor Mendel. Mendel, or Mendel, however you want to say it, Mendel sort of provided Darwin with the clarity of what he needed to fulfill his theories on evolution. And so these two fellows kind of worked together. They didn't work together, sorry, their ideas came together into what we now know as the common theory of evolution. So a big little summary of Darwin, we'll hopefully hit this more, but his boat was named the Beagle, and he sets out to get geological, geographical, and biological data and observations. And he did a lot of work. I mean, he kept meticulous notes and traveled for years, and was actually in religious school at one time and decided he had a propensity for this. And anyway, he came up with this theory that existing species, so not just new organisms, but new species of organisms, arose by what he called the descent with modification from ancestral species. And this is what we believe today to be true. Now, as we've seen, other theories have been proven to be false, but this one has been kind of, has not even come close to being disproven. So it's a really, really, really strong theory. And natural selection is at the basis of it. It's basically saying explains the mechanism of this evolution. We'll talk more about it, and you'll hear a lot about natural selection. And I'll leave it there for now. But again, he was a little bit more about a species, but he wasn't the only one that was, again, talked about this descent with modification, the big part of the theory. Natural selection, kind of the driving force. And natural selection is basically this. And I'll give the example that we can see every day in antibiotic resistance. You have several bacteria that are floating around in your belly or something, and you give an antibiotic and you kill off 4 out of 5 of them. Now, in reality, there's a lot more. Let's say that 5th one that you did not kill had a mutation that made it resistant to that antibiotic. It's the only one left in that population to reproduce, and so it's going to pass on that mutation, and by naturally selection process, that one is going to be the dominant trait going forward, or the trait that's expressed, the one that was resistant, and that's how you get an antibiotic resistant bacteria. You don't get the antibiotic does not cause a mutation and then cause the resistance. It's a response. It's just dumb luck that there was a mutation that existed that made it survive the exposure to that antibiotic. And so we can see evolution in a laboratory setting and all over our laboratory world with islands and our natural world as well. But Darwin wasn't the only dude that was thinking about this. There was this man named Russell Wallace, who was also coming up with these same theories. But again, Darwin was onto something. He knew what was going on, but he couldn't quite explain everything. He needed heredity. He needed traits. He needed to understand genetics. And Mendel came along a little bit later in 1866 that sort of solidified some of these ideas of Darwin's. And Mendel is kind of known as the father of genetics. Now, Darwin, evolution, but it's really hard to separate genetics and evolution at times, because what's in the gene pool, the the alleles or the genes that are involved from a genetic perspective influence evolution. So anyway, back to Mendel. He was worked with peas. He did these classic studies. He showed with data how traits are passed from offspring in predictable ways. He came up with the terms heterozygous and homozygous, or those concepts, and dominant, recessive. And he learned that each trait is controlled by a pair of genes that actually separate during the formation of egg and sperm, but come back together when the egg and sperm fertilize and create you, or whatever the species is. And so these traits are controlled by a pair of genes. Sometimes one gene is stronger than the other, but there's always a pair, and they that are expressing that same trait. They're working in pairs. And so his work was largely unknown until 1866, where he published it, and he's kind of, again, considered the foundational father of genetics. We could talk a lot more about that, but there are things that are classic Mendelian, which is dominant recessive. So those are the basics of what we learn when we teach genetics about homozygotes and heterozygotes and that sort of thing. But the reality is there's a smear of gray area in between where genes behave uniquely or differently based on various factors of how well they penetrate and other things that are going on with their biochemistry, and also a little bit about, just the influences the environment has and such. So anyway, there's some terms that we really need to get straight right away, and they're in the introductory chapters, and you need to kind of solidify these. And so sometimes we talk about two different types of organisms, ones that have more defined nuclei and some that don't. In this case, for humans, we are eukaryotes, and we have a nucleus, and we perform cell division called mitosis. And when we form our sex cells, the ones that are going to help us reproduce, girls have an ovum, guys have sperm. When we do that, we call that cell division meiosis. There's an extra division because we got to get from a full set of chromosomes to half. Instead of 4, we want 2, because we want them to go into separate gametes so that we don't have a chromosome mess in our hands when we fertilize. So I hope this is review for many of you, but you need to get some of these terms straight. Mitosis, diploid, meiosis, haploid, what a gamete is. And so let's go over that, what a chromosome is, because it gets a little bit like, what's a chromosome, what's DNA, what are these genes, where are they all located, how do you tell them apart? Those things are legitimate questions. So mitosis is the term that refers to cell division that occurs. We need to repair and replace cells all the time. And I like to say repair and replace, repair and replace. That's mitosis. So our bodies need to heal themselves, and cells have a lifespan, and they die, and they need to be replaced. And that's what mitosis does. Chromosomes in this case are copied and distributed into new cells. So you always start with a parent cell, and you get 2 daughters that look identically the same as far as the chromosomes. They're identical to the parent. We use the term diploid to mean that there's that complete set. There's it's called 2N or diploid. It's a full set of chromosomes. When you get to haploid, think of the word haploid and half. That's when you would get to half the number. So if I'm an organism that starts out with 46 like me, the haploid number would be 23. The diploid number would be 46. Different organisms have different numbers of chromosomes in their genetic makeup. So like I mentioned, meiosis is the formation of those gametes. There's a reduction of chromosome number by half, and that's what we put into a gamete, an egg or a sperm, and that's intentional because when they meet back up again, they're going to get the full diploid complement. All right, so there's some advances in science when we used to just use the light microscope like you use in biology, but they at one point discovered how to make an electron microscope, and that really helped us understand chromosomes and what's going on. And there's another term that I'm going to start talking about, which is homologous chromosomes. And anyway, this all leads to the chromosomal theory of inheritance. And so hang with me, we're still sorting through some terms, but homologous chromosomes are basically the fact that you get one from each parent and they match up. They're going to contain the genes at the same locations, and they're homologous, they're the same size, they're just going to match up, with the exception of sex chromosomes. They're different. X's and Y's that are involved in determining sex are different. they're homologous in the sense that they kind of line up together, but they don't look the same. And they have, the Y has a bunch of missing stuff, which makes it interesting. All right, in cell mitosis, whoops, backing up a little bit, this is all about cell division, and there are distinct phases of cell division, and one of them is what's called interphase, which is the normal life of a cell. They have what are called homologous chromosomes, and they're not very organized in interphase. But this one, the blue one, let's say that came from your dad, and the red one came from your mom. And they're still homologous. They're just, they've been duplicated during part of the process. In order for us to go from mitosis, where we have one cell, and we're going to make two out of that one, we have to double everything and then split it up. If we want to make half of that, then we would split again and get into the halves, which is meiosis. But anyway, back to mitosis. So these are called homologous chromosomes, and you can see where they're pointing. When you've replicated them and you're in that phase of mitosis, it's called, these are called sister chromatids, and it's just another term. It's all chromosomal material, but it depends on which stage you're at in the cell cycle. Anyway, these are homologous chromosomes. There's 46. This is a male, X and a Y equals male. 2 X's would be female. This is chromosome number 1, 2, 3, 4, and keep going on and on and on. All right, and so this is how we get to the 23rd pair, which is the sex chromosomes, and you can see they're not exactly homologous, but they're close. So these are, chromosomes, and this is called, this specific chart is called a karyotype. All right, there's some other people that were involved with this, Walter Sutton and Theodore Boveri. They formulated this concept of chromosomal theory of inheritance, basically that the DNA is residing in the big things called chromosomes, and the inherited traits are controlled by the genes that are residing on these chromosomes, and they are faithfully transmitting information through the gametes, like we talked about, to maintain genetic variability. There are a lot of things that go on when we do this, when our bodies do cell division, especially in meiosis. In genetics and in general, variety is good. We want a lot of mixing up in the gene pool. And so there are postulations that say that there are, like let's say you and your partner are able to reproduce. Every time that you create a baby, there are 64 trillion different combinations of genes and things, genetic makeup that can occur out of that. That's why we're all very unique, but there are similar features, obviously, from our ancestors. But in general, you're pretty unique because there's a lot of shuffling around that goes on with chromosomes. All right, I got to get going because you can't listen that long. So some other genetic terminology: alleles, mutation, genotype, and phenotype. Spend some time trying to sort these out. Allele just is kind of an alternate form of a gene, and it's like you could have blue eyes, brown eyes, green eyes. Those are all the same gene that's going to code for eye color, but there's different variations of that, or alternate forms that will give you green, blue, or brown. Eye color actually has about 9 alleles, so there's different forms of that gene. A mutation is when you have a variation in the gene or allele in the DNA, actually in the sequence of the DNA. I'll show you a slide here if that will help. Mutations are good and then they're bad. Sometimes they're good in that they provide a source of genetic variation, which I talked about with antibiotic resistance. That's why we don't typically wipe out something completely, because there's usually some variation that will survive, and that gives me hope as a human. And the super volcano hits or something, somebody will survive somehow. But anyway, mutations can also be dangerously severe and cause disease and distress. And I have one that's pretty devastating as well, but I'm okay. But so they can be bad as well and cause disease. The term genotype refers to the alleles that you were given, what you were given from your parents, what versions of those, the genes basically. Genotypes are your genes and the specific version of those. The phenotype is what you look like, how it's expressed. And one of the things I'd like to start off with, which I didn't do justice in this lecture, is how important the phenotype is, because your body, everything that you see and everything you do that you can't see is done and controlled by genes. And they're usually making a protein to do that work. And so your eye color, your hair color, lack of it. All of these things come from the DNA in your nucleus getting transcribed and translated into a protein. So proteins are not just what you eat as far as chicken goes, but they're what your body makes to do a lot of work and express your genetics. It's very cool. All right, there's some descriptions about some organisms that helped determine some of these concepts of phenotypes and genotypes and mutations with Drosophila. The short version of the story, for lack of time, is mostly the Drosophila are fruit flies, and they have a red eye, but sometimes there's a white-eyed version, and that's a mutation, and we will talk a lot about mutations. But some organisms studying different organisms has given us a lot of insight into genetics. All right, so here's a fun picture from me. Chemical nature of genes. What is it that's actually controlling our genetics? Well, it wasn't until 1944, which I'm 54 now. I was born in 1964. So about the time of this picture, I was being born, but my mom was born around 1944, and they were just then. You think you've been hearing this over and over again since you've been in school, but it was in 1944 where they finally realized that it was DNA that was the carrier of genetic information in bacteria, not protein. They knew about proteins, they knew about DNA, but they didn't know where the genetic information was carried. And it's carried in the nucleus, in the DNA, but it's interpreted as a protein, and it's expressed because of a protein. So there was some additional research that provided some solid evidence of this. But the bottom line here is this field is very young. My mom was just being born during this year when they figured that out, which I find astonishing. All right, so what does DNA look like? It was originally, or not originally, but more exactly described by a team called Watson and Crick, and I believe they won the Nobel Prize for that in 1953, and they described it as a long ladder-like molecule that twists and forms to form a double helix, and it has 4 different nucleotide subunits. And this is critical to understanding how these proteins are made, because the instructions lie in your DNA, literally, to express a protein to make you and express your eye color, your hair color, how your kidneys work, whether or not you're diabetic, all of that. Not necessarily. You get my drift. So these nucleotides are contain what's called a nitrogenous base, and there's 4 of them that are mainly involved: adenine, guanine, thymine, and cytosine, and they typically form these complementary base pairs. So adenine and thymine are going to pair up and bond across that helix of the DNA. And guanine and cytosine will also bind. And anytime you see that, that's who's coming together and being attracted. When that separates, and we use that DNA strand to make RNA, there's another player involved that gets replaced for T, and it's actually a uracil, and that's what is really transcribed into a protein. Is that messenger RNA has a different sugar base, and it has uracil in there instead. All right, anyway, so here's a very important sign. And I'm going to try to hit this over and over again this year so that we get it and understand it. But there's a central dogma of genetics and how genetic information moves in an organism. It's coveted and contained originally in the nucleus of a cell in the DNA, and it's protected in that nucleus. It's protected. But it's of no use if we don't use it to make a protein to keep. It's not a one-shot deal, folks. Your eyes aren't brown or green or whatever they are because it was just done at once and it was done. You constantly have to supply that pigment. If you don't, there'll be a change in eye color, literally, and I've had one. So there's things that can change. But you start with that DNA, it's coveted in the nucleus, but then, we want to keep it there, always have that master file so we know exactly what we always want. But we need to make these proteins. And the way we do that is by transcribing that DNA into what's called RNA. RNA is a single strand, and it has been made from that template of the DNA, and it's going to move out of the nucleus and go into the other part of the cell where all the machinery is, and it's going to find a ribosome, and it's going to make a protein. And so the flow of genetic information is that from DNA in the nucleus to RNA that moves outside into the cytoplasm and makes a protein, is used to make a protein. And again, proteins are not just what you eat, they're made by our cells to express our features, function, structure, and characteristics. In essence, proteins are the way our DNA expresses itself literally. Anyway, So what does it look like? Just a couple things. These are the base pairs. This is the sugar group, and these are phosphate groups. And so that's deoxyribonucleic acid. It's got an acidic nature. It's called a nucleic acid. So these are there's different bonds that are formed between the sugars and the phosphates and the base pairs. But these are what are called complementary base pairs, and these things are also called nucleotides, okay? And yeah, the sugars, so when it's made into RNA, it has a slightly different sugar, but don't worry about that now. This is the structure of the ladder. You've all seen that before. Here's a picture to kind of get you going a little bit. Really, it's all the same thing. A chromosome is DNA is, yeah, it's the double helix, all that. It's kind of the same thing. But what happens is if you start from small to big, so if you take the ladder and if you take the ladder and open it up, you can see these base pairs in here, these nucleotides that are going to bond with each other and kind of form that twisted ladder. But as the ladder twists, there are some molecules or some structures called proteins called histones that compact it and form it into, kind of wrap it up and condense it into material that's at one point called chromatin, but eventually will become your chromosome. And so the genes that we've talked about and alleles are located on chromosomes, but they're really sections of DNA, and they have a location that we use to identify them within the long strand. If we stretched the chromosome out, it would be a very long strand, and it wouldn't fit in our cell. So we have to condense it with these histones and wrap it all up. But so chromosomes, genes, DNA, they're all really basically part of the DNA, but it's just different ways we think about it as far as organization. They're bound tight into what's called chromosomes. There are locations on the chromosomes that contain areas or genes that do coding of protein. All right. And so RNA, like I talked about, is called ribonucleic acid. It's chemically similar to DNA. However, it's just single-stranded. It contains a sugar ribose, and one of the nitrogenous bases is uracil instead of thymine. All right. We talked about this already, but DNA contains information in the nucleus. Inside the nucleus, it is transcribed. So we make a copy. We're like, okay, we're going to make a recipe kind of thing. We're going to keep the master. So we start making the protein technically in the nucleus, but we don't finish it there. We just get the organization of the information into messenger RNA, and it becomes a single strand. It's complementary, and except that uracil is there instead, and it's going to move from the nucleus into the cytoplasm, and it's going to find a ribosome. So transcription takes place inside the nucleus. Translation occurs out in the cell cytoplasm with a ribosome and some other organelles. All right, translation is then where that messenger RNA takes the code. It's got the recipe, and it's going to take it to the ribosome, and it's going to crank out the protein of interest that we want. And that's kind of read in 3 chunks of the code. It's called, they're called codons. That is actually technically called translation. And when you put these 3 groups together, one group of 3 nucleotide sequences from RNA, messenger RNA, and eventually another RNA, but let's not go there. Those become what are called amino acids. When they're in groups 3s and you string together the amino acids into groups, they eventually make proteins, and they're cranked out by the ribosome. We'll do this more, but this is an introduction. So this is the DNA. This is the template strand that has the genetic information. These are the ends of the DNA called 3' and 5'. So we'll get to that a little bit later. Transcription, I can put a line right here and say transcription occurs in the nucleus, but when we want to translate it into a protein, it's got to go out to where the working organelle is, which is out in the cytoplasm of the cell. It's going to be translated on ribosomes into these amino acid groups. They're based on those 3 letters, and that is going to string together and make a protein. All right, we've already talked about that. There's basically 20 different amino acids that are found in proteins. And what's really interesting about this is, and we will hit this a lot harder when we go into this stuff, but the genetic code is redundant and not ambiguous, and it's got some built-in fail-safes. Very cool thing. So we'll talk about those amino acids. But right now I want to move a little bit into like what goes wrong when one of those base pairs is substituted for something like that. In other words, mutation. Again, we assemble these proteins. Let's just go on from there. One of the largest group of proteins that we have are enzymes, but there's a lot of others: hemoglobin, insulin, collagen, actin, myosin, all the things that are going to pigments. They're all proteins. I teach a pathophys class, and some of my students tease me that I always blame it on the proteins. What's usually the fault of a protein? Because it's either inflammation or protein. There's kind of a couple things that I always am saying is that, anyway, and autoimmune diseases, these are things that are commonly themes that are coming out of my mouth, but proteins just function and run everything about your body. So, and a big group of them are called enzymes, which are biological catalysts. They help us do our biochemical reactions in our bodies, and they are very important proteins, as you can imagine. All right, so let's talk about mutations. If you have a change in, let's go back here for a minute. If you have a change in one of these 3 letters, so these 3 letters right here, AUG, will code for a specific protein. I'm sorry, a specific amino acid. Methionine is this, valine and serine and leucine. These are amino acids that are directed to be made based on these 3-letter codes. If you mess up, and DNA messes up one time when it's replicating or something, there's what we call a mutation, then you have some problems, or you have some good things that could come out of it. But problems include, an example would be sickle cell anemia. It can alter the ability of the protein to fold properly or to function properly if we have a mutation. And sickle cell anemia is not deadly, it's just a nasty disease. It's debilitating. It affects the blood. It makes the blood cells, instead of flat, round, disc-like, that are deformable and can move through capillaries, they become sickle-shaped and cannot work well. And it's the result of one little substitution for one little letter during the making of an amino acid. Anyway, it's a mutant form of hemoglobin. It results in misshapen blood cells, and it leads to a lot of problems. But it's, again, if you look at it here, this is normal hemoglobin, the DNA to messenger RNA to amino acid, which is the building block of a protein is an amino acid. So here is where you have the problem. In a normal hemoglobin, you're supposed to have glycine here, and you have valine instead, and it's the error is right here with this T and the A. At this spot, there should have been a T. Now, sometimes there's a mutation and it still translates into the same amino acid, so it's silent. It's no big deal. But in this case, there's a substitution that is thrown off at this amino acid production, and it makes sickle cell anemia. One little mess up. That's a picture of a sickle cell. These are normal red blood cells. That's a sickle cell 1. All right, we're almost done, folks. So a lot of information that comes out of knowing stuff about your DNA. Recombinant DNA technology gives us rise to the biotechnology industry. Basically, what we've been able to do is figure out how to manipulate DNA by cutting. We figured out where in that DNA strand we can cut, and we use enzymes to make, they're called restriction enzymes at one point, endonucleases, but later we call them restriction enzymes, and we can cut into the DNA and insert what we want into that with ligands, another type of enzyme, and we can get an animal like a bacteria, or an animal cell like a mammal cell or a bacterial cell, to express the protein that we want and make it into a drug. It's really cool stuff. That's what recombinant DNA technology is all about. We've been able to do this with crops as well, and transgenic organisms. There's a lot of bad news about genetically modified organisms, but in a way they've been allowed us to feed more people. And so we got to debate the good and the bad, but there's a lot of genetics in plant science and agriculture and in medicine, where I was spending my time. There was one thing that was done that's not really, it gets talked about a lot because it's cool, but we were able to clone a sheep. Dolly was cloned in 1996. And so there was a nuclear transfer. There was no sex involved or recombination. We took cells from Dolly and implanted them, transferred them into an egg with the nucleus removed, and produced a genetic identical offspring with desirable traits. Some of these transgenic animals, we can do this with and utilize their DNA to make things for us, like drugs. But this is a picture of Dolly, and she gave birth to a cloned copy of herself. But the thing is, genetically speaking, they will be the same. However, they will be exposed to different environmental things and may not have the same personality or the same experiences or the same contribution to the environment on their genetics. And so there will be differences, but they genetically are like twins, so to speak, identical twins. All right, so these have been beneficial to help us get anti-clotting protein from milk, to help with that in clinical trials and therapeutic proteins like I was talking about. All right, there are about 10 million people that suffer from genetic disorders. I think it's higher, actually, depending on what we count as a genetic disorder. There's a 3% chance of a newborn that will have a genetic anomaly. Biotechnology sort of gives us hope, and we have been able to treat over 100 inherited disorders, and there are still some ethical concerns to be resolved, but I'm a big believer in it, and so yeah, it's very cool. And the other thing is there's legal issues around patents and things like that. The Trump administration currently is trying to make it faster for drugs to go to market, which I'm for, but there's a lot of safety concerns, and I understand all of this stuff. But one thing that he wants to do is get kind of remove the ability to patent something that you discover from a genetic standpoint. I don't like that because people will spend their lifetimes working. It would be like if you spent all your life trying to create an invention and then you didn't have any protection with that. But his intentions are not bad, but the people that do all this work and devote their life deserve some reward for it, in my opinion. All right, some of the branches of studies that come out of this are genomics, proteomics, and bioinformatics. And it's just studying the human genome or other evolution of genes and genomes in different organisms, studying proteins and studying, bringing it all together in databases with bioinformatics. Very cool stuff. All right, one thing that we used to talk about, there's different ways to study genetics, and you could look classically forward and study mutations that occur naturally, or you can reverse that and sequence the DNA for a gene of interest and try to determine its role and function by knocking out an area and see what happens. So it's kind of interesting. We'll talk more about knockouts. And anyway, the book talks about model organisms that we use to study genetics. We can't always do them on humans because we don't have quick turnover. We're not easy to grow, and we don't have short life cycles, and we don't produce a lot of offspring, heaven forbid. So they use organisms like Drosophila and other mice and other things that have a quicker turnover time. Drosophila is a fruit fly, which is a mouse and fruit fly right there. Other model organisms are viruses and microbes and different plants. So there's the Elgin's nematode worm is often used for neurological studies and things. So E. coli is in your gut, and yeast. So there's more. All right. So yeah, let's take a break here for a minute. I gotta regroup. Whoops. All right, a couple more slides. I wanted to see where we're at. I know I'm taking a lot of your time, but so some of these organisms, model organisms, are used to apply information to treat humans, and we can transfer genes between species. And we've learned some things from this approach to study some neurological disorders like Huntington's disease and Alzheimer's. We'll talk more about Huntington's, but it's a nasty one. It's an autosomal dominant one, which again we'll talk about. Here are some model organisms: E. coli and Drosophila and elegans and the mouse, to just different diseases that they're studying. I would, this is just for fun and information. Well, it's not fun, but you get my drift. All right, and so there's been, it started with Mendel, and it's been rapidly growing as a field, but it's still very, very young, but full of potential, folks. Just one of the coolest things out there is the study of genetics. So I'm really excited about this class. Here's a bit of a timeline. It was around here, this time in the 70s and 80s when that biotechnology burst was happening, and then in the 90s, the Human Genome Project, and we're starting to learn how to apply that information. So Watson and Crick, this is about the time that my mom was born, but this is Mendel here, but way back when, before BC, we knew enough to manipulate genetics to kind of get a hardier plant or to change wheat and feed the world and that sort of thing. All right, so there have been many Nobel Prizes awarded, and there's still some sensitive issues related to prenatal testing and discrimination and ownership of genes and patents and how to safely do gene therapy and not, infect with viruses and just be safe, and some privacy issues. So even as of late, the 23andMe and DNA things that you can send out for your ancestry, they've actually used that to incriminate somebody. So there's some fun ethical issues with this as well. So there you go. That's a little bit of an introduction, and we'll just end the meeting at that, and hopefully you'll enjoy the lecture.